Hi, listeners. Thanks for joining me today for episode 751 of the People Behind the Science podcast. I'm your host, Dr. Marie McNeely, and I'm looking forward to rebroadcasting our interview today with Dr. Stacey Harmer, originally published on October 3rd of 2016. Listeners, Stacey studies different biological rhythms and the circadian clocks within organisms that create and maintain those rhythms. Circadian clocks can be found in a variety of organisms, from bacteria all the way up to humans. And Stacey is particularly interested in understanding why plants have circadian clocks, how those clocks work, and what aspects of plant physiology and development those clocks control. And in our conversation, she talks more about her life and her research. So listeners, get ready for this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. Today, I am thrilled to introduce everyone here to Dr. Stacey Harmer. So, Stacey, how are you today?
I'm doing very well. How about yourself?
I'm doing quite well. Thank you very much for asking. And thank you for joining us on the show today.
Excited to be here.
Excellent. Well, Stacey, let me take a moment to introduce you to our listeners and introduce our listeners to you. So listeners, Stacey is a professor of plant biology in the College of Biological Sciences at the University of California, Davis. She received her bachelor's in biochemistry from the University of California, Berkeley, and she was awarded her PhD in biochemistry from the University of California, San Francisco. Afterwards, Stacey conducted postdoctoral research at the Scripps Research Institute in La Jolla before joining the faculty at UC Davis, where she has been now since 2002. Now, Stacey is the recipient of the American Society of Photobiology's New Investigator Award, and she was selected as a Chancellor's Fellow at UC Davis. And listeners, Stacey is here with us today to talk about her experiences in life and science. So, Stacey, we can't wait to get to know more about you and your life, both inside and outside the lab. So, can you start by telling us what you do with your time when you're not doing science?
I think it's really important to have a life outside of the laboratory because, as I'm sure you and listeners know, science can be very stressful. And for me, I actively try to find things that allow me to stop thinking about my work. I do yoga and I do a lot of road biking. And those, for me, are two really good ways to kind of get outside my usual mental state and think about either nothing or think about different things. And then the other nice way to relax that I really enjoy is cooking and eating. And I like the latter, especially when my husband does the cooking because he's a very accomplished cook.
Wonderful. Does he have a particular food specialty?
Yeah, we were talking about this the other day because he always is at a loss to describe what his favorites are when people ask. I think what he came up was rustic urban.
Ooh, I like it. Well, I must say I'm a fan of eating. I'm less of a fan of cooking. So if my husband could cook for me, that would be great. Well, Stacey, wonderful to hear more about your life outside of the laboratory. And I think you're absolutely right. It's so important to have these other aspects of your life to change your perspective on things, to get you outside of your lab mindset for a little while. So, Stacey, we do want to talk about your laboratory and your research next. So can you tell us, how do you describe what you do to someone who might not be familiar with your field?
So when I talk to someone who's not in my area of research, I often will describe myself as a chronobiologist. And what I mean by that is that I study different biological rhythms. When you say biological rhythms, sometimes people don't quite know what you're talking about. It could mean a lot of different things. It could mean the cell cycle. It could mean life cycles. Mm-hmm. But for me, I'm particularly interested in the circadian clock. And the circadian clock is something that you find across all kinds of organisms, plants, animals, fungi, cyanobacteria even. And these circadian rhythms are generated by an internal circadian clock that is an internal timer that generates 24-hour rhythms in physiology or behavior. And one of the things I really like about this topic is that people can relate to it. Most of us have experienced jet lag, right? So we always know that when our internal clock is out of sync with the external environment, that things don't feel right.
Right.
But people are often surprised to learn that plants have these internal clocks as well. So I've really enjoyed understanding how the clocks work in plants. And then I'm also really interested in understanding what they do for a plant. Why do plants have them? And what aspects of physiology and development do they control?
Absolutely. Well, I think this is fascinating. When I think about these biological rhythms, I often think of them being controlled by the brain, but maybe that's biased because I'm a neuroscientist. But thinking about plants, where would you say this control is centered? Because they don't tend to have the brains, quote unquote, that we think of.
Yeah, absolutely. And actually, the point you make about the brain controlling rhythms in animals, that's partly true, but it's not exclusively true. Mm-hmm. So for both plants and animals, you can isolate single cells, and those cells have 24-hour clocks running in them. They're intrinsic, that they're cell autonomous. And when I first learned about that, it seemed like science fiction almost. How can a single cell keep track of time? And yet lots of organisms have that ability. So plants, as you said, they don't have that brain coordinating behavior and physiology across the whole organism. And it's an area of debate right now in the plant field as to how much influence do particular tissues have on other tissues in terms of circadian clock function. We used to think that it was essentially every cell was being sort of independently entrained by the environment. So the timer in those cells being set by light-dark cycles or by temperature cycles. Mm-hmm. But now there's some evidence that maybe different parts of the plant, like the shoot apical meristem, is influencing clock function in other regions of the plant as well. But it's still not quite sorted out.
Definitely. Well, this is really cool research, Stacey, and I'm excited to dive more into your projects as we chat more in this conversation. But I do want to touch a little bit on motivation. I think this is really important for scientists who often find their to-do list ever growing. How do you stay motivated? How do you continue to get it all done? And do you maybe have a favorite quote or saying that keeps you going?
Well, it's interesting you ask, how do you get it all done? Because I don't think you do get it all done.
Right.
At least I don't get it all done. And you have to, I think, just kind of learn to accept that your to-do list will never be finished. Mm-hmm. But in terms of motivation, a quote that I really like is one from Albert Einstein that says, if we knew what we were doing, it wouldn't be called research, would it? And what I like about that is it points out that in many areas of science, you figure things out as you go along. Like you have a plan, of course, and you try to follow that plan as you go, mm-hmm. but things aren't going to turn out the way that you expect them to, and you're going to have to deviate from that plan, and you may abandon it altogether and come up with a new hypothesis that you're going to follow up on. So I like that. It's okay to be a little bit uncertain. That's just part of the process.
Absolutely. And I think you're really highlighting this unpredictability in research. And I think that's something that people maybe take for granted. They assume that scientists know exactly what's going to happen just because we've formed a hypothesis, but we're very often wrong.
Exactly. Right. And that's when things can get interesting.
Absolutely. Well, thank you for giving us a window into what motivates you and one of these fantastic quotes. But I want to talk a little bit next about some of the people who might have motivated or inspired you along your journey. So, Stacey, can you take a moment to tell us about one of your mentors or role models?
So my background in training was in biochemistry, so in a pretty different field from the plant biology that I do now. And I've been really excited learning about the people that have been pivotal in the area of plant biology. And I find that there are some really inspiring figures there. And one person that I find particularly impressive is a scientist named Winslow Briggs. And he's a scientist at the Carnegie Institute at Stanford University. And one of the things I love about his career is that he's made really important contributions to plant biology over a very long period of time. So back in the 1950s, he did some physiology revealing how this plant hormone called auxin is involved in directional growth of plants towards light. And then 40 years later, in the 1990s, his lab was the one to clone the blue light photoreceptor that's important for that directional movement. How many people have significant contributions to science over 40 years of time?
Yeah.
And he's, in fact, still doing really amazing science at the age of 88. And so his longevity and the way that he has been able to change the kinds of things that he does, right? Because our tools and techniques have changed incredibly over the span of his career. And he's always been able to kind of adapt to these new approaches and take advantage of them. So I find that really amazing and inspiring. And also, he's just a really great guy. He's done all these impressive things, but he's quite modest. He's happy to talk to people about science and not concerned about letting you know how important he is. And he really, I think, conveys his love of biology to all the people that he interacts with. So he's a really great person.
Certainly. Well, I think you selected an excellent role model, someone to look up to. But I'm curious, do you aspire to be one of these people who's still working in your 80s, 90s?
That's a great question. I used to think so. I used to think that I would always be in the lab. But now I think that maybe at some point I might want to retire and put some of my energy doing other kinds of things, maybe more travel. No time soon, but maybe I will stop doing this job before I am forced to do so.
Sure. Well, that sounds wonderful. Thank you for sharing one of your mentors. But now, can you take us back to the beginning, Stacey, and tell us where your journey began? Can you tell us what is the first memory you have of being interested in science?
I used to think I was going to be a veterinarian. I'm not sure that that qualifies as science in the way that you're thinking. But I've always liked being around animals and liked being outdoors and have been attracted to biology in that standpoint. So I've always had that, I guess, emotional attraction to biology. But actually, at some point, I discarded the idea of being a veterinarian. And I thought I was going to become some kind of a physical scientist when I went to college. And I think the reason for that is in high school, I just did not enjoy my biology classes very much. Mm-hmm. So therefore, I thought that I didn't like biological sciences. In retrospect, it's probably because my teachers weren't very inspiring. Because when I started taking college courses in biology, I realized that, in fact, I was fascinated by understanding how living things work, especially how they work at a molecular level. So I had to kind of change my ideas about where I was going to go pretty early on in college.
Gotcha. So then can you tell us, what was your next step then? You got into college, you were starting to realize you were interested in biology. What happened next?
I had the opportunity to work in the lab of the late Daniel Koshland when I was an undergraduate. And just through advising and reading about things, I had learned that if I wanted to know if I really liked biology or not, I should probably get some experience doing it myself. Mm-hmm. And he was happy to have me work in the lab as an undergrad. And that was a really fabulous experience. He was a very successful scientist. When I was in his lab, he was the editor of Science magazine. He was quite well recognized.
Sure.
But he was never too busy to talk to the undergraduates that were working in his lab. So he would talk to me about my project and things that I might try. And he was excited about my successes. So that was really encouraging to me. And that experience of working in his lab for 2 years really convinced me that I wanted to pursue a career in research. So that was a really pivotal experience there.
Definitely. So then once you decided to pursue this career in research, how did you go about picking your PhD program and deciding where you wanted to go next with it?
Right. So I knew I wanted to go to graduate school. I was pretty undecided as to what kind of a topic I wanted to do. So I ended up looking at schools that had rather broad programs where you didn't have to...
You could put off the decision.
Exactly. Yeah. Exactly right. And so I applied to a bunch of schools, interviewed at some, and I was really excited by the feeling of intellectual engagement that I found at UCSF. People seemed really, both the faculty and the current students, they just seemed really engaged in what they were doing. They seemed to be having fun, which wasn't true at all of the schools that I interviewed at.
Right. Me too. Yeah.
Right. And so it just seemed like a place where I could have fun too, as well as get a great education in science. And for me, that was a really good choice. I had a great experience at UCSF. And I focused there on B-cell signal transduction, because I was really interested in understanding how cells signal to each other, how information gets passed along and results in a change in biological fate. And so that education was really just a great introduction to biochemistry and signaling.
Gotcha. And then you went on, as I mentioned in our introduction, to do postdoctoral research at the Scripps Research Institute. Can you tell us a little bit about your experience there?
So although I really enjoyed my grad school experience, by the end of it, I was a little bit frustrated by the system that I was working in, because I was studying B cells that had been growing in culture for many years, and I wasn't studying an intact organism. And I just decided that I wanted to be able to study a biological process more in context of an organism. And I also wanted to be able to use genetic analysis to better understand how things worked. In particular, how when you lose function of particular genes, how does that alter the thing that you're interested in? So I definitely wanted to change my area of research. And I took quite some time, I'd say about a year and a half, just sort of looking at diverse research topics and different kinds of labs to try to find something that I was excited about. And that ultimately led me to the lab of Steve Kay at Scripps Research Institute, where they were just on the cusp of using genetic approaches to dissect the circadian clock in plants using this model plant Arabidopsis thaliana. And that turned out to be a really great decision for me.
Fantastic. And now here you are today at UC Davis. Can you tell us a little bit more about something you have going on in the lab right now that you are so excited about?
Okay, so yeah, a few years ago, we decided to expand our focus beyond Arabidopsis thaliana, which is People call it kind of the fruit fly of plant biology. It's a very powerful model organism, but it's not a crop plant, and it doesn't have a lot of the different aspects of physiology that other kinds of plants have. And we decided that we wanted to study how the circadian clock affects plant growth in the natural environment. And furthermore, we decided that a great model system was going to be studying potential clock regulation of solar tracking in sunflower. And most people are familiar with the fact that young sunflower plants move their stems and leaves from east to west during the day to follow the sun. But most people don't realize that at night they reorient the whole plant, the stem, the apex, the leaves, from west to east in anticipation of sunrise. And that seemed like a process that the clock was probably going to be involved in. How does the sunflower plant
know, right?
Know. Yeah. How does it know when the sun is going to rise and what direction it's going to come up in? And we were able to show that the clock was involved and, in fact, required for that nighttime reorientation. And so now we're trying to get at the molecular level to try to investigate how the clock interacts with light signaling pathways to give us these really interesting and distinctive growth patterns.
Certainly. Well, this is fascinating. And do you think this is something that's unique to sunflowers? I mean, perhaps it's easier to see in sunflowers, but do you think it also happens in other plants?
Yeah, so that's a great question. And I think what you said is correct. It's very obvious in sunflowers, because that stem shows that very dramatic back and forth movement. But it turns out that many plant species actually do show this kind of solar tracking, either of leaves or of flowers. And we suspect that related mechanisms are going to be involved. And just sunflower makes a nice model system because you have this big stem that you can dissect and extract proteins and RNA and so forth and try to understand the mechanism in that plant. But we think it's going to apply to other species as well.
Certainly. And I think the significance extends a little bit beyond just the physiology of the growing plant. I did a little bit of reading in your work and saw that there are some implications for pollinators as well. So can you talk about that?
Right. So another thing that people often don't realize about this process is that as the sunflower plants mature, their heads, they stop moving. So by the time you see a sunflower with the yellow petals and so forth, that plant has stopped stem elongation, it's stopped its daily growth rhythms, and now it's fixed facing east. So both dawn and dusk, you'll see those fields of flowers with the flowers facing in the eastward direction. So we were interested in why that was, both on a physiological level. Why do the flowers face east rather than random directions or west? And also from a more ecological standpoint, does it matter for the plant that the flowers face east? And we found that the circadian clock is involved in controlling how responsive these plants are to light. So if you stimulate a plant with directional light in the morning, it bends very efficiently in the direction of that light. But if you do the same thing in the afternoon or the evening, it's much less sensitive to light. And that's something called circadian gating, that you can give a stimulus at different times of day and see a different degree of response. And that's actually very common. You see it in plants, as we did for light responses. You can see it in animals for responses to different chemicals. So there's lots of examples of doing the same thing at different times of day, but seeing a slightly different result. But we found that the plants are more responsive to light in the morning. They're insensitive to light in the evening. And so the eastward orientation is because the stems are sort of slowing down their growth over time as they age. And that afternoon sunlight is just not a strong stimulus for them. So they don't bend very far west as they age. But then at night, they bend back towards the east.
Gotcha.
So eventually, as the stem growth ceases, you end up with that frozen orientation in the eastward direction. Does that make sense?
Yeah.
Yeah, okay. And so then we went on to investigate, was there a difference in terms of the plant's success in facing east or facing other directions? We found that pollinators, particularly bees, very much prefer the eastward-facing flowers to flowers that were manipulated to face west. And we speculated it was because the eastward-facing flowers warm up faster during the day than the west-facing flowers. We could show that. And that when we used artificial heaters to warm up west-facing flowers so they were the same temperature as the east-facing flowers, we found that now pollinators liked those warm west flowers almost as much as they liked the undisturbed east-facing flowers. So temperature played a big difference there.
This work is amazing, Stacey, and I think we could talk about it all day, but I would like to hit some other topics. So one thing that I do want to chat about are some of the challenges that you face. I think we've talked about a lot of the exciting aspects of your career and how you got to where you are today, but I think we haven't yet touched on a lot of the struggle, the challenge, the failure that happens to everybody in science. So Stacey, do you have a story of one of these incidences that you can share with us today that kind of showcases these challenges and tells how you got through them?
Yeah, so when I was a graduate student, I went through a phase maybe of a year or two where pretty much nothing seemed to be working. And so some of this was just the usual kind of experimental problems that everybody encounters. A piece of equipment breaks mid-experiment, or I made a mistake setting up the protocol, stuff like that. But some of it was just because biology didn't work the way that I thought it would. I had a hypothesis, wasn't the correct hypothesis, and so the line of experiments wasn't going to be fruitful. And it was really frustrating because I was working quite hard, but I just didn't have very much to show for it. And I think particularly because it was kind of early in my graduate career, it was a big contrast from being an undergraduate just taking classes. And when I was taking classes, if I worked hard and studied hard, I got better grades. I did better on exams. So there was a nice correlation between effort in and product out.
Sure.
That just wasn't happening with my experiments. I could be working really hard and just not getting much positive feedback from that effort. And that was really difficult.
A lot of graduate students go through this period where it seems like everything's falling apart and everyone kind of has different strategies to get through these low troughs in their experience. So what helped you?
Well, I wish I could say that I had a magic bullet. Instead, though, I just kept plugging away. I had a variety of different lines of experiments that were going on, and eventually a couple of them started to give me interesting data. And those things, of course, then became the basis for my thesis. So that's kind of one piece of advice that I like to give students now is don't have all your eggs in one basket. Start out at least doing a few things at once. And if something's not working, just keep trying. So for me, this was actually, in retrospect, a really valuable experience because I learned that although my experiments can fail, that doesn't mean that I was a failure. Just my experiments were not me.
Absolutely. And I think it's also points to like the idea you were saying of the correlation between sort of productivity or output and how hard you were working. I think that's something that continues on into your faculty days too. You can slave away at a grant and not get the grant and feel like you failed.
Well, yeah, right.
So
that's also depressing.
Right. It's good to get used to that kind of thing though, early on in grad school.
Right. And just kind of have the experience that I'll get through it. Things will get better.
Absolutely. Well, we don't want to just dwell on the difficult times, Stacey. So can you tell us next about one of these happy moments in your scientific career, a success, whether it was a big one or a small win?
Yeah, so I was thinking about some recent things that have been positive and exciting. And one thing I thought I would mention was that we've been trying to understand for a number of years now the function of a gene that we identified originally, because when you mutate that gene, the circadian clock runs with a different pace in Arabidopsis plants. And the identity of the gene was interesting because it's highly conserved across all kinds of organisms, all kinds of eukaryotes, humans, mice, zebrafish, yeast, etc. So the fact that it's highly conserved suggests that it has some important and perhaps conserved function in these diverse organisms, but nobody has reported what it does, like on a molecular level. So it's a tough protein because it doesn't have any recognizable domains. You don't look at it and say, oh, it's a kinase or oh, it's a transcription factor. So we've been plugging away, trying to figure out what the protein does on a biochemical or molecular level. And we still don't know what it does, but a reanalysis of some old data has made us think that its main function might be in repairing damaged DNA. And we're following up on that possibility now. And that still is going to leave us a ways to go to try to understand what the protein does. But that insight, which of course I still don't know if that's true or not, but that hypothesis is leading us to new types of experiments that we hadn't previously considered doing. And I'm hoping will kind of lead to a breakthrough in this project that's been really a tough nut.
Interesting. So what was going through your mind then when you started seeing these pieces fitting together that this might be involved in DNA repair?
Well, it's actually very exciting because then you kind of go back and think again about all kinds of phenotypes and experimental results. And again, this may just be a house of cards, but being able to say, then if this is the case, I'll find a similar phenotype in the literature and some other mutant, and then going through that literature and finding that that's true, that's a really powerful feeling. It's very exciting.
Absolutely. I think these are the aha moments that scientists get so excited about.
Right. But then you have to remember not to get too attached to your hypothesis because it may be wrong.
Right. And I think if you go in with that bias too, you start to put everything in to fit the pattern of the hypothesis that you have, and suddenly everything makes sense when really it might not.
That's right. But it's fun. Building that house of cards is a fun process too.
Absolutely. Well, thank you for sharing one of these successes, and congratulations on this potentially very exciting discovery.
Well, we'll see. I hope so.
Yes. Well, Stacey, we talked a little bit about your life outside of science as well, and some of the things you like to do with your time. And I know one of the things that I am trying to fit in more and more, in addition to reading all the literature, keeping up with the papers in my field, is trying to read more for fun. So I do bug everyone we have on the show to give us a book recommendation. So, Stacey, can you tell us about a book that you've read, whether it's a science book or a non-science book, that you have really enjoyed and think we should read?
I would strongly recommend a novel that I read just in the past few months that's called Measuring the World by Daniel Kellman. And this, it seems a little bit unlikely, but it's a novel, so it's mostly based on biography, but the author's taken some liberties with historical fact. But it recounts the lives and interactions of two very famous scientists, Alexander von Humboldt and Carl Gauss. So Humboldt was a famous 19th century scientist explorer, and Carl Gauss was a famous mathematician of the same era. And there's such contrasting figures. So Humboldt is a very bold explorer who travels all over the world, climbing volcanoes, going across glaciers, measuring everything that he can find, like literally measuring it, taking just huge numbers of records. And then Gauss is a stay-at-home. He's afraid of travel. He doesn't actually do experiments. He does thought experiments, but he's got tremendous insight and intuition and comes up with the basis for actually many theorems that people proved after his death. And so you've got this master of inductive reasoning and a master of deductive reasoning, and Kellman imagines how they would interact with each other and how their lives would kind of reflect on each other. It could be quite dry, but it's actually really funny. It's a really funny satire. So I highly recommend that book.
Well, Stacey, I am going to have to add this to my list. I love the contrast that you're drawing between these two historical figures. So we will definitely put this on our reading list. Listeners, you can find it there as well. And Stacey, I think one of the things that is a great opportunity as a scientist that we haven't chatted too much about yet are some of the opportunities you get to travel. So can you take a moment to tell us about your favorite travel experience for science, where you were and why it was memorable?
It's a little bit hard to pick one favorite because, as you said, you do get to go to a lot of great places as a scientist. And one of the other things that makes travel rewarding is once you've been in this field for a while, you start to know people all over the world. People you say you trained with as a graduate student or as a postdoc end up kind of scattering to different places, which is sad because you don't see them much anymore. But it's nice because when you go to those places, you have people you can visit with.
Right. Every trip's a reunion.
Yeah, exactly. And so that's great. And one of the trips that I took that I quite enjoyed was a visit to Sweden to the Umeå Plant Science Center, where we had a friend who has a position there. And my husband is also a scientist, and they very kindly invited both of us to give seminars there on subsequent days. So this was one of those trips where both me and my husband got to travel together. So that was great. We really enjoyed our trip at the Science Center, both scientifically and also socially. But what for me was quite memorable is we had a chance to visit Sweden, a place that I hadn't really been before. In particular, we visited a national park called the Abisko National Park. Mm-hmm. It's north of the Arctic Circle in the northern tip of Sweden. And we went in September, and it was just an amazing experience. The park has forests of birch trees, and they had turned this just an amazing golden color. It was super striking. I'm someone who likes to hike and do things outdoors, and this was just a completely different kind of wild environment than the one that I'm used to. So it was really memorable and striking.
Well, this sounds phenomenal. Were you able to do a lot of bicycling while you were there in Sweden too? I know you mentioned that's an interest of yours.
No, we didn't do any bicycling.
Really?
You can only do so much on one trip.
That's true. That's true. Well, I think a challenge too is whenever I find myself going somewhere interesting for a conference is you try and squeeze a vacation in there around any lectures and other things that you have going on. So you find yourself with a really crowded itinerary. But
yeah, exactly. You have to strike a balance.
Absolutely. Well, thank you so much for sharing this trip to Sweden that you had. It sounds like a phenomenal experience. And one of the things that I do also love to chat about are some of the people in science. You mentioned being able to travel, you're able to meet people all over the world, but also reconnect with people who you've worked with previously. And I think the people in science are part of what makes it great. And I know there are stereotypes out there about what scientists are like and what being a scientist is like, but we love to kind of break some of those on our show. And one of the things that I like to highlight are just some of the quirky traditions that scientists often come up with, or just the funny memories and things you do when you're together. So do you have a story of yours, Stacey, that you can share that really highlights this human element of science?
Here at Davis in my department, there was a time of maybe, 3 or 4 years where every summer we would have a cook-off competition.
Ooh.
And so what this meant was that the different labs in the department would take turns preparing what started out as just a coffee break with snacks for the rest of the department. And then to keep things interesting, there was a panel of judges that would taste the different offerings from each lab. And then at the end of the summer, a lab would be rated as the winner and would receive a fabulous trophy. So this was kind of a fun tradition. It got people together. It allowed people to be creative in terms of the kinds of things that they would bring forward for the cook-off. And one of the things that I thought was really a very clever entry came from a lab that does a lot of genetics. And they decided that their theme for the cook-off was going to be a genetic screen.
Interesting.
So they decided they were going to make chocolate chip cookies, many different batches of chocolate chip cookies. So one batch was made following the recipe, adding all of the ingredients. But in each of the other batches, they left out one ingredient. So they made one without the salt, they made one without the baking powder, and so forth. And then all of the trays of cookies were set up and you could go through and taste them and see, okay, what happens to a cookie if you don't add the salt or the baking powder or the eggs?
I like it.
Yeah, so it's a great illustration, I think, of the concept of genetics. And also it was kind of, from a culinary standpoint, it was a pretty interesting example as well. What happens without the leavening agent in your chocolate chip cookies?
Right. All of them, I'm sure, had different states of looking appetizing versus not looking appetizing.
It was not the most delicious of the efforts.
Right. Were there any of the screens that ended up tasting better than the original, though?
No.
Nope. Not even close.
Not even close. We actually had to finally discontinue this tradition because people's competitive instincts got a little bit too strong. I mean, it started out as a coffee break and it ended up as kind of an elaborate early lunch. Mm-hmm. And the judges were being treated to white tablecloth and table service. And
oh, wow. It just kept escalating.
Exactly. So it was fun, but I think we took it just a little bit too far. So then it became too extreme.
Yes. It's funny. A lot of the scientists I've talked to have competitive traditions. You don't realize how competitive your lab mates are until some of these things start. And then you're like, oh my gosh.
Exactly.
Well, a fantastic tradition to share with us today, Stacey. And I love this idea of incorporating some of the scientific elements within this baking contest. I think that's awesome.
The other thing that was kind of fun is often people, since labs are often quite international, people would have a theme where every lab member would bring something from their culinary tradition. So that was really fun too.
Absolutely. Get to try a lot of different types of food. That's fun.
Right.
Well, wonderful to hear more about these lab traditions. And I think when you're together with your colleagues, you talk about the current research, but I think it's also fun to chat about these big questions in science, whether it's in your field or just across science in general. And think about, if you had anything, what are all the possibilities of questions you could answer? So I want you to tell us today about the big question that remains in your mind, Stacey. So if we were to suddenly give you everything, funding, staff, technology, feasibility, whatever you wanted, what is the one question you would want to answer with it?
There's so many ways to go with this question, but I think if I really had unlimited resources and everything at my fingertips, the research question we would want to solve would be, how do you reverse global warming? How do you get greenhouse gases out of the atmosphere? Because that is something that would make a huge impact on the world and would be really an amazing thing, which, of course, with my background, I don't have the training to do it. But if I had unlimited resources, I could collaborate with people that would.
You could share your resources.
Yeah, exactly.
I like it. Well, this is definitely one of the big questions facing all of society today. So very important to be thinking about and trying to figure out little ways that all these different scientists from a variety of fields can work together. So great question that you would dedicate your resources to. And now, Stacey, we talked a little bit about some of the people you've encountered across your scientific career. And I think a lot of these people are great sources of advice when you're in need of advice throughout these different points in your career. So can you tell us, is there one piece of advice that really stands out as just the best or most helpful that you ever received that you can now pass on to our listeners?
A member of my thesis committee in grad school was Mike Bishop, and he's famous for this quote, "It's your career." And you can sort of take that in multiple ways. You could see it as kind of maybe a cautionary statement. Mm-hmm. Okay, you take the vacation, but it's your career.
Right. I wouldn't do it, but
yeah, exactly. But on the other hand, you can also see it as a reminder of the freedom that we have. Many of us working as scientists, we have a lot of options as to what we work on, what problems we want to approach, how we're going to approach them, what kind of exact experiments we're going to do. And that freedom, I think, is really precious. And so I think that also kind of feeds into this aspect of it's your career. So it's, I think, a reminder of the responsibility that you have, but also the freedom that you have when you're a scientist. So I really like that advice.
Absolutely. I think that's a great little kind of Post-it note to stick on your computer to remind you, okay, what grant am I going to write? It's my career. I have to decide. Put the reins in your hands.
Right or not write the grant. It's your decision.
Absolutely. So Stacey, is there any other last piece of advice or a last note of inspiration that you want to end our conversation on today?
I often will talk to students who are finishing up their PhD, thinking about what to do for a postdoc. And sometimes they're very focused on, well, what's the technique or what's the particular question that they think is going to get them a job after their postdoc. And, of course, we need to think somewhat strategically, but I think it can be a mistake to get too fixated on that. And so what I try to tell people is instead of trying to guess what the next big thing is going to be, because you're probably going to be wrong, whatever you guess, but instead try to find a problem that's really captivating. So if you're excited about your job, you're going to work harder, you're going to do a better job, you're going to enjoy it more. And I think you're ultimately going to be both happier and more successful. So try to put the time in, the hard work of figuring out what problems you really want to devote yourself to. And I think that's very much time well spent.
Absolutely. And I think the more excited about your projects you are, the better you're able to package them and kind of sell them to these places you're interviewing at.
Absolutely. And as you mentioned, writing grants, if you don't think it's important and interesting, how are you going to convince other people that it is?
Absolutely. Well, fantastic advice to leave our listeners with. Stacey, now, can you tell them how they can reach out if they want to learn more about you and your work?
Yeah, they can send me an email. My email address is slharmer@ucdavis.edu, and that's a really good way to get in touch.
Excellent. Well, listeners, if you have any specific questions for Stacey, definitely reach out and ask her. And Stacey, thank you so much for joining us on our program today.
Thank you. It's been a lot of fun.
Well, it has been a pleasure to have you here. And listeners, as always, a pleasure to have you with us as well. We'll see you next time on another episode of People Behind the Science. Your voyage to explore the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to chatting with you next time on People Behind the Science.